The idea behind the pump is this idea that while I'm working out, if I do something, can I send more blood flow to my muscles, right? And therefore cram more things into those muscles, and will I look like I have, or will I actually have larger, more defined muscles, right? Like during my workout or in some kind of period of time right after my workout, okay? And so associated with all of this are supplement products like this. So this nano vapor stuff. When we moved into my lab, it's been 8 years ago now, we found a sleeve of this stuff called nano vapor that had been left in there. The guys that were in the lab before us, the faculty members, did a lot of research on supplements. They were not always above board with the money they were taking in various things, and that led to them no longer working here. But they left some, and I have it downstairs still. If anybody has ever gotten something like this, the stuff written on the package is the most hilarious thing you've ever seen, okay? It makes your crazy grandma's Facebook like thing look, I mean, it talks about like, don't open this and disturb the powder or it will explode. And like you will have like, you know, the world's most massive gains and this super rush, you know, it was hilarious. And so we all had like vowed that when my 3 master's students all graduated, that we were gonna save it and we were gonna open it up and like, you know, go and like, you know, do cheers and have a drink of whatever the stuff was when they graduated. And Robbie's still here. The other 2 are long gone. But the idea in this is that it has stuff called nitric oxide, okay? It has something that's a precursor for nitric oxide. As you all will learn, nitric oxide is a vasodilator. So what that means is that it opens up blood vessels. It opens arterioles, and that, in theory, then allows for the pressure inside those arterioles to drop, allowing more blood to move through them faster to a particular area. We'll talk all about how we control arteriolar diameter and how that affects where blood flow goes at any point in the thing. And so what they tell you is, oh, well, look, we've got this stuff, right? It's got nitric oxide in it. It's going to open up all your blood vessels going to your muscles, and you're going to get this massive, huge, like, pump, and you're going to be all swollen and look like super ripped, plus more blood flow. You're going to get all that protein that you're taking that's also in here, all that creatine that's also in here. It's all going to get to your muscles easier and faster and better. Bullshit. Every last bit of it, okay? There are things in here, probably it's caffeine. They won't tell you like, oh, it's this proprietary energy blend. It's always caffeine. That's all that it is, okay? Or it's a precursor on one side of meth, basically, or ephedra, okay? Because ephedra is illegal unless you get it from Canada or someplace. Anybody? So I had another master's student who I guess Marson was American, but he went to school in Canada. His father worked at a university in Canada, and so he would go home at Christmas and he would smuggle ephedra across the border because you can buy it in Canada. And so Marson was a trip. But you can get it on the black market. I'm sure that's the least of the things we should be worried about that Cameron Combs is taking. But the idea is that this is going to give you more of a pump that will supply more blood to your muscles, more nutrients to your muscles, and allow you to have bigger muscles at the end of the day. You'll recover faster, you'll get bigger gains, and all of this. Total junk. This is the idea of bro science in its most kind of boiled down, purest essence. There's real science in this. Nitric oxide, which in here it's not nitric oxide, it's something called L-arginine. You can just go to Walmart and buy the L-arginine and get the same effect. L-arginine gets turned into nitric oxide. That's real. It dilates blood vessels. You take this, it may actually vasodilate some blood vessels, okay? That's good if you're a person that can't do that under normal circumstances. Well, as my guys in the cardiovascular lab could probably tell us, unless you have atherosclerosis, unless you're a smoker or something, every one of you can perfectly vasodilate the blood vessels or your arterioles going to skeletal muscle during exercise. Because if you couldn't exercise, okay? And so you jam a whole bunch of protein in there. What does it matter if more protein goes across those muscles? You have to have done something in the muscles. The muscles have to take it up. Like the lack of blood flow and things to the muscles is not the limiting factor in why your muscles will get bigger or not, or why you're going to work out during, get one more rep or not during exercise if you have normal function, as most people do. So this is one of those things that this is what supplement companies do. There's real science here. There's stuff that actually does things. And they tell you that, but what they don't tell you is that, well, yeah, it may do that, but the thing that it does isn't really that important. The caffeine is the most important thing in here. The only thing in here that probably really helps a person is the caffeine. We'll talk about that. Okay? Anyway, somewhere in my lab, Ryan, you can kind of go find it, in a cabinet deep and buried is the nano vapor. It's in a sleeve. I think it's grape flavored. But you've got, I meant to bring it. You read the package. It's hilarious. So this is a pre-workout. Okay. Other things that are the most complete waste of your money in the entire time: pre-workouts and protein powder. Okay. Now, if you've got the money and you want to buy it, that's fine. Rather than having the pre-workout, go to Walmart and buy caffeine pills. You can, for $2, you can buy like 100 of them. That's all that's in there in the pre-workout that's going to be, that's doing the things. All right, so there's our growth science for the day. All right, so let's talk about my most favorite thing in the entire world, muscles. Okay, and the neuromuscular system, kind of how skeletal muscle is innervated by the central nervous system, and how the signals from the central nervous system are going to tell the muscles to turn on and off in a particular fashion. Okay? One of the things that we need to learn and appreciate in class, much more so I think than as an undergrad, is this idea, and you will see this across all of the systems that we talk about, is that the structure, the anatomy of the system is going to play a very, very key role in imparting the function of that system. Okay? All of the anatomy that we have outside of like our appendix or something, this stuff did not happen by accident. Okay? There's millions and millions and millions of years of evolution from lower animals into us on why this is set up the way that it is. Okay? Now we can argue till we're blue in the face about how we've removed kind of all natural selective pressure on ourselves right now. Other than maybe I'm not going to flee the hurricane that's coming in some ways. But so let's start with this. Okay, how does anatomy impart function on a muscle? Muscles have one job only. Okay, I would argue that this is the most important thing that happens during all of exercise physiology. So what do muscles do? Somebody can just read off of your slide there. Muscles do one thing. They generate force, okay? I'm talking, I need force, okay? I'm gonna do a squat. Oh yeah, okay. Haven't done very many of those in a while, but I wore my stretchy jeans on that particular day, okay? So muscles generate force. That's all that they're there for. Their entire structure and anatomy, okay, is designed to allow us to pack as many actin and myosin proteins in as small a space as possible so that we can generate as much force as possible in the size or the space that is available to us. Okay? Then, those muscles are going to be arranged in a particular way to allow us to move because we're bipedal organisms. Okay? The anatomy or the cellular anatomy of my bicep is not that different from, say, my quads, right, or my hamstrings or my glutes. But the whole muscle arrangement, the direction of pull, its pronation, which we will talk about in things, is going to be a little bit different from upper body to lower body muscles. Okay, the things in your eyes are going to have a much faster fiber type. They can contract much faster than, say, the muscles in your calf or the muscles in your diaphragm. Because if they don't, then you get shit in your eyes all the time and you can't see, and that is bad. Okay? So generating force, that's going to kind of help. Our anatomy is going to help us do that for the specific location and task that that body segment is doing. Right? Motor neurons, right, or the nervous system, they have one job, really two. It's kind of all wound up in one thing. Okay? They are there to tell a muscle when to turn on and when to turn off. Okay? It's the gas and the brake pedal in your car, basically. That's what the motor neuron is there for. And so when we see where motor neurons are located, what their structures are, how they gather signals through their dendrites from the surrounding neurons, things coming down from the brain, how their axons, which is going to seem really wild, okay, the way they have their axons set up, they're so far away, but these things are going to be, okay, set up to allow us to very quickly turn a muscle on, turn it off. And not just turn it on, but turn it on to just the right precise amount of force that we need for a particular task. Okay? If Ryan generates too much force in her fingers, she smashes through her laptop while she's trying to type. Okay? Every time you do something, your brain makes an estimation of about how much force that it needs to perform that correctly. And then it gets feedback while you're doing it, and that's how you're able to actually write and type and walk and run and talk. Some of us can't like walk and chew gum at the same time, but it makes an estimation and it turns multiple muscles on and off at just the right amount of force every time. Okay? And then in real time, you're running on uneven terrain or something, or you're me and the ginormous St. Bernard tries to yank you across the thing. It allows you to then make adjustments almost in real time as well. We'll talk a little bit about how the nervous system is set up to allow for all of that to happen. And to me, every time we do all of this, I'm always amazed that we're able to do anything, right? Because so many muscles, so much coordination that's required. But the structures are set up to make that the case. Okay? All right, so why are muscles and motor neurons important for exercise? Well, exercise requires coordinated movements involving multiple muscle groups and this varied amount of force that's going to change from moment to moment based upon whatever it is that you're doing. Okay? Hundreds of muscles may be contracting simultaneously. Okay? They're contracting bilaterally. You've got one foot that's up, you've got one leg that's back, you've got to have the right arm doing something. This arm is doing something while this foot's doing something else while I'm shoving this person away. I'm thinking about kind of like a Heisman pose or something here. Okay, I'm really missing football right now. But if these things weren't structured and put together in the way that they are, these things aren't able to happen. Okay? And so just keep that in mind as we look at all of this. Why do I make you learn all of this crazy, ridiculous stuff that's in the stupid Grey's Anatomy book? Okay? Well, because if it wasn't that way, we couldn't move the way that we do. Okay? And we are very unique as bipedal organisms. We move differently than almost any other organism on Earth. That has imparted us some good things and some not so good things, right? So there we go. All right. That's kind of the big general overview of all of this. Okay? Some basic facts about skeletal muscles. And most of this is just kind of for your edification. There's about 650 muscles in the body, so you got a lot of muscles. Okay? Each muscle, think of each muscle as a Russian nesting doll. You guys know what a Russian nesting doll is? Yeah? No? Christian, you know what a Russian nesting doll is? No. You don't? Okay. So who was on there and they know what one is? See if we can, somebody explain it to Pearson what a Russian nesting doll is.
Isn't it like just a big doll and you have like other
And how do they look in comparison to each other, right? They're exactly. So a Russian nesting doll would be like something that's the size, it would be like this, and then you split it in half, and inside is an exact, sort of slightly scaled down replica of my thing of paper towels. And then you open that one, and there's a little smaller one, and a smaller one, and a smaller one, okay? Mussels are Russian nesting dolls of these kind of decreasing in size or increasing, depending on which end you want to start on, of these little tubes, okay? And so there's these thousands and thousands of cylindrical cells that are muscle fibers that are going to lie in parallel to each other and lie sometimes longitudinally with each other, okay? Things that make skeletal muscle unique. As you guys will see, each muscle fiber in your muscle. So muscles are made up of thousands, hundreds to thousands of fibers depending upon the size and the function. Each fiber is a single muscle cell, okay? Muscle fibers are some of, if not the largest cells in your entire body. If you let me cut your leg open, okay, I could pull out an individual muscle fiber. You could see it with the naked eye. Might even be a couple centimeters long, okay? I can't like pull out a cheek cell or anything. You can't see it without a microscope, okay? So these muscle fibers are single cells, but they're giant. And so because of that, they have some unique properties to them. There's a reason bacteria aren't the size of this room, okay? If you have any background in biology or cell biology, there's a thing called surface area to volume ratio. It's the same idea of why it's always the offensive and defensive linemen that have trouble with heat stress on the football team, okay? But cells can only get so big before when they have one nuclei and whatever their capacity to make protein and things is going to be. They can only get so big and be supplied with the proper stuff that they need by that single nucleus. That's why bacteria are small. They have one nucleus. They can only get to be so big. Okay? Because muscle fibers are so big, they have adapted. They have multiple nuclei in them, which makes them very, very unique. Okay? Additionally, those nuclei, I'm probably not supposed to use this, but we're just going to hope for the best. They gave us our own, they probably gave it to you, they gave us all our own like set of like markers and things, and because I'm not used to this at all, I just leave them in my bag in my office. But anybody else having trouble, like anybody use the hand sanitizer that's like right outside the window? It smells so bad. There's a lot of alcohol. I'm like, it smells like a margarita. Do they make that with tequila or something? But so you've got a muscle fiber and it looks like this, right? And so the nuclei in muscle fibers are very sort of interesting because they sit out here along the edges, okay? That makes them unique. In most cells, the nuclei will be, kind of here in the middle. And that's going to become important when we look at muscle fibers under a microscope at determining how old is the fiber, is it an old fiber, is it a new fiber, and a couple of things, because the nuclei are not always in the same place. Okay. They may vary from a few millimeters, very small muscles in your eye, to nearly 30 centimeters, okay? Fibers may be 30 centimeters long down here in your legs. So, I mean, that's like a foot or something. Foot, yeah, something like that. Okay, so very, very big. As it relates to our pump idea, muscles are basically water and protein. They're mostly water. Okay. In this instance, we'll call it sarcoplasm or cytoplasm, depending upon how you want to go about doing your cell biology. Okay. That's going to play some important roles on some other things because I can force more water into my muscles by having you work out, right? We can make Callie get up and just do squats for about 10 minutes. And we could stuff more water into her leg muscles and she would appear to have bigger leg muscles. And then about an hour while it goes away. Okay, so the proteins are mostly actin and myosin and then a few other ones, but those are going to be the two two big ones that we really didn't like. This is the idea of the Russian nesting doll, okay? You have whole muscle, so just imagine my bicep, okay? Bicep is here. If you were to cut open my skin, peel back my subcutaneous fat, peel back the connective tissue, then you would see that my whole muscle is made up of multiple of these bundles of muscle fibers called fascicles. They have connective tissue. You open that up, you get down to the individual muscle fibers, okay, which are individual cells, okay? You then can cut open the cell, and inside the cell are going to be what appear to be these other smaller tubes that are made up of protein, which are myofibrils. And if you pull out and look at one myofibril, you will see that inside that myofibril, its individual proteins are going to be aligned in a way that it looks like something which we're going to call a sarcomere, which we'll talk about in a minute, okay? And then the sarcomeres are made up of proteins, okay? The way all of this works, though, is something happens at the protein level. One protein pulls on another protein. That pulls the whole sarcomere. So the sarcomere moves. As individual sarcomeres move, they pull and the myofibrils move. Then that makes the fibers move, which make the fascicles move, which then makes the whole muscle move. So when I do a curl like this, okay? Please no one laugh at my lack of, sort of bicepness in the midst of all of this, okay? But I do a curl. There are millions of interactions of actin and myosin proteins going on in my bicep. Those proteins are generating the force that allow me to move, okay? They move, then there's going to be millions of sarcomeres that are all moving these tiny infinitesimal amounts. And when you group them all together then, because each successive one of these is surrounded by connective tissue, like rubber bands, it's stretchy, so they move, they pull the connective tissue. That connective tissue pulls on the next bigger structure, which then pulls on the next piece of connective tissue, and it pulls and it pulls and it pulls, and eventually then the whole muscle moves, that pulls on the tendon, and then that's going to attach down here, and it's going to, if I get enough force, it's going to pull my arm, okay? That's what's happening when you do muscle contraction. Every time you move, what's really happening is, on a microscopic level, you're getting millions and millions and millions of interactions of actin and myosin proteins. And the protein called myosin, the myosin grabs the actin and it pulls it ever so slightly. And then it lets go, and it moves back and it attaches again. And then it pulls, and it lets go, and it attaches. And it does that boom, 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 millions and millions of times. That's where the force comes from. That's where the movement comes from. It's from these tiny, tiny little things. But we see it as the whole muscle moves, and then that makes everything else go. Okay? So again, it happens here. This is all set up to allow us to pack as much actin and myosin protein into this area that we can possibly have, surrounded by connective tissue, so that when it moves, it can transmit that force, and then that lets us move and do things. Okay? Does that make sense? This is the part where if it doesn't make sense, it makes sense to me, but it's at times it's not going to make sense to you. And I'm going to go too fast, I'm going to get excited, and blah, blah, blah. You got to tell me. Okay? So that's kind of how and why all of this is going to happen. Okay? All right, so we talked about the connective tissues that are going to be there. All right, so let's now try to go a little bit more microscopic. And so I kind of told you guys the end already. We don't teach you guys. So when I was a grad student, if you were going to teach a class, we had a class that we took as grad students about how to be a good teacher and those kinds of things. And one of the first things they'd tell us was, start at the end. Tell people the important thing that you want them to know at the end first, and then kind of work backwards from there. So I've already told you guys what's going to happen, right? The actin and myosin interact, they generate force and movement, right? It kind of gets transferred all the way up. So what we're going to do now is figure out in the kind of between whole muscle and actin and myosin, are there some other structures that are going to be involved that are going to help us turn those actin and myosin on and off and allow them to function properly, okay? So what you see here, okay, this is a cross-section cartoon of a single muscle fiber, all right? The outside of the muscle fiber, so this is one cell, it has a cell membrane like all other cells. That cell membrane is a lipid bilayer, okay? And it's going to have proteins interspersed in it. It's going to have receptors interspersed in it. It's going to have glycosylations all over the place and other things that we may talk a little bit about later on in class. But there's a membrane. There are going to be holes in this membrane that are connected to like channels, okay? And so what I want you to know is this channel is called a T-Tubule, right? It's a channel, it's a tube, however you want to think about it, okay? But just imagine that I've got my water bottle, my Diet Dr. Pepper here. This is my muscle fiber, okay? And kind of at various points interspersed here, I've taken and I've just sort of pressed in and I've opened this up and I've run a little straw down into it. That's the idea of what a T-Tubule is, okay? Its job is to take a signal from outside here and bring that signal down in and around where the actin and myosin are so that we can actually get it to do what we want it to do. Okay? T-tubules are wildly important. We will get to situations where we talk about something called fatigue, right? I say, Callie, get up, do squats, okay? If you guys won't talk one day, we'll just do squats until somebody talks. Okay? I can say, Callie, do squats. At some point, she's no longer going to be able to do any more squats. One of the reasons why is that we will have messed up the ability for T-tubules to transmit signals. So it's really important structure. Okay? So we have sarcolemma, T-tubule, okay, connected inside the cell, immediately adjacent to the T-tubule is this what looks like a web-like network. It doesn't really look like this. We keep getting better and better imaging techniques on what sarcoplasmic reticulum looks like, and it's actually not really like this. I don't want to freak you out because I've always seen it this way for like 30 years, and I see new pictures, and I'm like, that's not it. No, that's not it. You're like, that's the world I'm living in? No, I don't like that. It makes me very uncomfortable. They're connected to sarcoplasmic reticulum. Okay? Sarcoplasmic reticulum is, they're just like closets or storage sites inside the muscle fiber. And they store calcium. It's their only job, store calcium. Lots and lots of calcium, okay? And so what's going to happen then is we're going to get a signal, it's going to go down the T-tubules. The T-tubules are then going to tell the sarcoplasmic reticulum to open the closet door and let the calcium. And that calcium is going to tell us to do some other things, okay? And then the signal is going to stop, or we're going to suck the calcium back in, and voila. Okay, but that's kind of what's going on there. Other things inside a muscle fiber. We're going to have these mitochondria here, here, here. All fibers have mitochondria. Some fibers will have more mitochondria than others. We will talk a lot about why that's going to be the case. Okay? Muscles that you use and contract more often than others will tend to have more mitochondria. We can specifically do certain kinds of exercise training in order to make the mitochondria in our muscles go up. More mitochondria, more ability to make ATP aerobically, which is going to be a good thing. You guys will see, probably not today, but next week, that we need the ATP for the actin and myosin to interact with each other. So we've got mitochondria here, so there's going to be ATP and energy available in very close proximity to these proteins. Okay? We talked about the nucleus. They're going to be kind of on the outside. They're going to be there. That's going to be the place where you want your muscles to get bigger, or you damage them and you need to make new proteins to make them grow or to fix them. That's going to come from the nucleus. Okay? Other things that muscles have that some other kinds of cells don't they have a thing called a satellite cell. Okay? Satellite cells are muscle-specific stem cells. Okay? Muscle-specific stem cells. Who can tell me what a stem cell does? Anybody know?
They can create new cells.
They can create new cells, yes. Any particular kinds of cells they can create, Megan?
I'm sure there is. I just don't know.
Okay. Anybody else know?
Stem cells change to whatever is around them so that they can kind of quickly heal up. They very specialized very quickly.
So stem cells tend to be undifferentiated cells that, in theory, have the ability to become any kind of cell in the body that you need. That's why they have such wild potential from kind of a medical standpoint. Okay? And so lots of people now, you guys may have even had it somewhere. I think we did it with Ellis, but they can, when babies are born, they can take some of the cells from the umbilical cord, some of the blood from the umbilical cord, and they can just like save it. And there are stem cells that are in there. And in theory, then you've got those saved so that in another 20 or 30 years, we keep saying in like 20 years, we've been doing that my entire life. At some point, we'll figure out how to tell those cells that are your cells to grow you a new liver or grow you a new lung or something, right? So that when you need a transplant, you don't have to find a donor. We've got your own, right? And so, anyway, but you have stem cells in your body. You have muscle-specific ones. These satellite cells are muscle stem cells. They can only really become muscles, but they play a really, really, really important role in muscle hypertrophy, when muscles growing and getting bigger, and muscles repairing themselves after they've been damaged, okay? And so we can do certain things to maybe make these work better or make them not work at all, and that can have some effects on your potential for muscle growth or muscle repair. That's why I just want to bring that up. We will come back to it later on in class. Okay. The other thing to keep in mind about the sarcolemma that's really, really important, okay? This is a thing that imparts function on skeletal muscle. The sarcolemma is excitable, okay? It's your friend that's just so pumped to be like outside and ready to go, okay? Not that kind of sarcolemma. In this instance, excitable means it is capable of transmitting or generating electricity, okay? So in many ways, your sarcolemma works like a nerve, okay? It can transmit action potentials, it can transmit electricity. This is really important because we're going to use electricity as a signal to turn the muscle fiber on and off. And electricity is in some ways the best signal. Why? I'll ask my engineer. Why is electricity good as a signal? Any ideas?
I'm really
Not sure?
honest.
Okay. Back in the day, way back in the day, like way like older than me, before there were phones, okay? We would send messages on like little teletype things, right? I forget what they were freaking called. Oh my God.
Morse code.
Not, it would be like Morse code, but it was a, I'd like text my wife and be like, what's that old thing called? Anyway, but wires would be strong, you know, like you could send a message from here to New York or something, and it would take seconds, right? Because you're transmitting electricity along those wires, and you would sort of type out your message like in Morse code or whatever, and then they would receive it like a telegram. That's what it is, a freaking telegram. So it's a telegraph system. God, I'm losing my mind. It's all the lack of sleep. Why would we use that? Because electricity moves really, really quickly. It goes very, very fast, okay? Much faster than, say, like the Pony Express would. So back in the day, you could write something out, give it to somebody, and the mail would try to deliver it, Pony Express would deliver it, or you could do the telegraph and get it there like practically immediately. Now we just, you know, text somebody, and I don't know, it goes signals off somewhere, off of like 14 satellites and around. I don't really know. We're all in the matrix anyway. Who cares? So that's the good part about the sarcolemma. Okay, the sarcolemma allows us to conduct electricity, and so because of that, signals can move across it very precisely and very quickly. Which is going to mean when I touch the thing that's hot, I can tell that muscle to contract and pull my hand away very, very quickly. Or Kelvin's coming at me, he's going to punch me, and I duck out of the way and run. Okay? So, all right. That's something to remember about the sarcolemma. I've got stuff in here about the basement membrane. The basement membrane is pretty cool. People do a lot of research on the basement membrane now. It is, in essence, the anchoring structures in those cells. Okay. The basement membrane is made up of a huge number of proteins and things, all right? Maybe one of the ones that you guys have heard of the most is a protein called dystrophin, okay? If you have, say, muscular dystrophy, you lack or you have a mutation in this dystrophin gene, depending upon your type of muscular dystrophy, this protein isn't made correctly, and therefore the scaffolding and structure provided into your muscles by the basement membrane, by this dystrophin, falls apart. And so people that have muscular dystrophy tend to have very small muscles. Their muscles get damaged really, really easily, okay? And so we are learning more and more kind of almost every week about kind of what happens in the basement membrane, and does it have a potential role in hypertrophy? Maybe. Does it have a role in kind of preventing and recovering from muscle damage, like when you're sore and things? Seems like yes. Should we put collagen into there? Kind of what are the things that we can do kind of to supplement this kind of stuff? We learn more and more. It's probably beyond the scope of class, but I did just want to bring it up that it is this thing that is there. Okay, and what it does is it really kind of anchors the muscle fibers in some ways to each other, but then also to, like, see here, this is this extracellular matrix. It's anchoring them out here to collagen. It's going to help us anchor them to tendons. Okay? So some interesting stuff going on there. All right, we talked about T-tubules. We talked about the sarcoplasmic reticulum. It always kills me. The end where the sarcoplasmic reticulum makes contact or comes into very, very close contact with the T-tubules is something called the terminal cisternae. I don't really care if you guys know this, but they always like to ask this question. Kelvin will appreciate this. I think I asked Callie to go, and she didn't, but usually in the ACSM quiz bowl, they always ask something about where is calcium located inside of a muscle cell. And you have to say it's in the terminal cisternae of the lateral sacs of the sarcoplasmic reticulum. And I roll my eyes because everybody just says it's in the sarcoplasmic reticulum, like y'all are being super kind of persnickety about things. But that's what this is, and that's why I put that on there, because Kelvin will appreciate this. He was on our quiz bowl team last year. Even though they lost, it was okay. We hadn't lost in a couple of years, so it's okay. The team that beat us, or both of the teams that beat us, their advisors and their school are the ones that make up the questions. So I think there's a little bit of nefariousness that's going on there. So it's okay. Kelvin, you want to weigh in, Kelvin?
Yeah, I think so. And there's also the judges that were a little not with us, because there was one question that was very controversial, and I asked about it, and they're like, nope, not it.
I know. So anyway, we can, on a different day when we have more time, we'll talk about this. Usually what would happen is I would encourage all of you to go. So the Central States American College of Sports Medicine Conference is usually in, last year it was in Tulsa, or outside of Tulsa, and we usually go. Grad students, you guys can go, and there are scientific presentations. If you have data of your own, you can do a poster, you can do presentations there, but they have an undergraduate quiz bowl. So every school in the region can bring a team if you want, and if you win, which we have won several times, then they pay for your team to go to the national meeting and compete in the national quiz bowl. And we almost won. We won the national quiz bowl like 2 years ago. If you win the national one, then you can get like real cash and things. Anyway, I had a point to all of this. I don't really know what it is. That's kind of what, that's the terminal cistern. They like to ask about that. All right, so things that I do want you to know because they are in some ways more important than the terminal cistern. Okay. Here we have, this is sarcoplasmic reticulum. This is the membrane along this right here is the T-tubule, okay? So imagine you've got a cell that runs like this, or I'll try to get out of the way so Ryan, you guys can see. My muscle fiber is running like this, right? Somewhere out here is the sarcolemma. I've got my hole. It's going to open into my T-tubule. It's going to run all the way down in there, and then kind of butting up next to it is going to be some portion of my sarcoplasmic reticulum. Okay? These three things you are going to need to know. They are really, really, really super important. Okay? So, on the T-tubule side of things, there is a receptor called the DHP receptor. DHP stands for dihydropyridine, which is just describing the molecular structure of the receptor. Okay? It is a voltage-sensitive channel. What that means, okay? What voltage-sensitive means is that it can open or close, or it will change its shape, basically do a conformational change, when it senses a certain electrical signal that goes by it. Okay? So what's going to happen is there will be an electrical signal that will be out on the sarcolemma. That signal will travel down into the T-tubules, and when it gets into the T-tubules, when it makes contact with the DHP receptor, it will tell that receptor to change its shape. Okay? The DHP receptor is in very, very close contact with a receptor on the edge of the sarcoplasmic reticulum called the ryanodine receptor, which is right here. Okay? The importance of the ryanodine receptor is its structure. It looks like the cap or a cork maybe in your wine bottle. If you imagine the sarcoplasmic reticulum as your wine bottle, the ryanodine receptor is the cork that is in the wine bottle. DHP gets activated and it pulls the cork out. It changes this little shape of the ryanodine receptor, which opens up, okay, opens up a pore, and it allows the calcium to then be able to move out of the sarcoplasmic reticulum. Then we've opened it, and then poof, the voltage goes away. This goes back to its original shape, puts the cork back in, and we've now stopped releasing calcium. Okay? These are really, really, really critical steps in the nervous system telling skeletal muscle to be able to contract, because we need the calcium to be released to get actin and myosin to bind up with each other. We'll get into that piece of that in just a few minutes. Okay, so that's going to be here. DHP and ryanodine are very well studied. Their structures are very well studied. We know a lot about these two kinds of things. Okay, there's also another structure on the sarcoplasmic reticulum. Okay. And if these are going to help us get a muscle to contract, then this enzyme here, called the calcium ATPase, or it is often called SERCA, okay? This one here is going to help us contract the muscle a second time, okay? So if these are going to get us to contract once, this functions. Once we've released the calcium, we get a muscle contraction. We close this back up, and then SERCA is going to function like a vacuum cleaner and suck all of the calcium out from the inside of the cell, suck all the calcium back into the sarcoplasmic reticulum. Okay? as a person that was at one point in my life not the cleanest person in the world, may I make a recommendation to you guys? This is an aside because I was talking about vacuum cleaners. They're outrageously expensive, but if and when you have the means, buy one of those Dyson like shark things. You'll believe, like, you'll never realize like how clean your carpet will be after all. Anyway, it's the thing. We bought one, but my wife made us get one, and every day we use it every day. It's the greatest like vacuum ever. Side note from Dr. White, it only costs $400. And I tried to get it for her as an anniversary present, and she was like, oh, hell no, we're just buying the damn vacuum. So, anyway, Circa functions like a vacuum cleaner, and it pulls the calcium back in. Okay? What we will see is that Circa is an enzyme, so it takes energy for us to run the Circa. It's basically a pump, okay? And it grabs calcium and it pumps it back in, or it pulls and sucks it back in. But it takes energy to do that, okay? So this is not a passive process, this is an active process. The speed at which Circa pulls the calcium back in is going to vary from muscle fiber type to muscle fiber type. And so experimentally in the lab, if we could do things in the lab, you could come down to my lab and I could basically zap you with electricity, make your muscle contract, and I can estimate your Circa by looking at some things about how quickly your muscle relaxes. Okay? So this is a thing. Fast twitch muscles have faster Circa. Slow twitch muscles have slower Circa. And the function of Circa is going to play some really important roles in the ability of those individual muscle fiber types to generate force. It's also going to play a very, very big role in fatigue, along with DHP and riot. Okay? I told you guys that we can mess up the T-tubules, and so what happens at times when you get fatigued and you cannot turn a muscle on properly, the T-tubules are really, really narrow, and so as you send all of these action potentials down through here, sodium and potassium are moving across them, and then we have to have a sodium-potassium pump to kind of reform our gradients. Because it's so narrow, the tube gets clogged up, basically, and it can't repolarize correctly, and therefore you can't generate an action potential. It will start on the sarcolemma, but it can't get down through the T-tubule. So then DHP can't get activated, and you can't release calcium, and that's why you can't generate force. Okay? So there's some things that are going to kind of go on there as well. They're unique to the way that these things are all going to be linked together. Again, structure imparts function. Okay? There's a reason that people with fast twitch muscle fibers are better sprinters. Okay? They can turn their muscles on faster, but they can relax them faster so they can get to the next contraction faster, and that's because their Circa works faster. Okay? So just kind of a couple of things. Am I going too fast? I think I'm going too fast. No. Have most of you, maybe other than Pearson, at least seen some of this before? These are not completely new concepts. Okay. Mitochondria make ATP. We'll get into how the membranes are going to be structured and what happens where and where the Krebs cycle happens and where the electron transport is. We'll get into metabolism. Those are mitochondria. Okay? Yay, mitochondria. They have their own individualized DNA. They were probably at one point single cell organisms that we just took up into ourselves. This is a electron micrograph of muscle fibers. The little things that look like maggots on here those are actually those are the nuclei. Okay. A few of them are probably also satellite cells. Under this kind of examination, we can't really tell. Satellite cells also tend to be out here on the outside. Mature muscle fibers have their nuclei, which we call myonuclei. They are going to be near the very, very edge. They're right under the sarcolemma. That lets us see all this stuff in here. This is actin and myosin. This is protein. So we're not taking up space in the middle of the fiber with all of our nuclei. They're all kind of shoved out here. It's going to help us generate more force and transmit that force better. We have many, many nuclei. Under kind of extreme magnification, satellite cells look like a nuclei, but they have this extra kind of double membrane that surrounds them and encases them to keep them from getting activated. Okay. We'll talk about protein synthesis and DNA to RNA to proteins much later in the semester. We'll be able to talk about how muscles get bigger, how they grow. Okay, satellite cells. If you really care, like, we can talk about all different kind of things. The way satellite cells get turned on, and there's particular signals that we can measure molecularly. Like we can look for proteins and stuff that get turned on. We can look for these immature muscle fibers that do things in like kind of single muscle fiber preps. You can look for those individual kinds of things. We don't really do that here in our department, but if you're interested, you may read a paper about it. I know a little bit. I know enough to be dangerous.
I got it. Supposed to. Yeah, it looked like the hurricane just kind of stared some storms out.
I have a picture of my daughter on the. It appears to be out in the rain on the playground at daycare. So, and she refuses to let us put like clips or rubber bands or anything in her hair. And she has my hair, and so it's just like plastered like straight down. It drives my wife crazy. There's baby Ellis. Okay. So I had a thing. Yeah, is it storming? That's not great because one of my bassets hates thunder and he freaks his shit out. And so if he's at home in the crate, he'll really not be in a good way. So, okay. Sucks. That's okay, Megan.
I was looking at the weather.
Just get videos. Okay. So we're going to move now from talking about muscle fiber specific stuff to talking about some kind of myofibril and like down to the sarcomere level things. Okay. So if here we have one fiber, we've been talking about the fiber, sarcolemma, T-Tubule, sarcoplasmic reticulum. Inside the fiber, we're going to have all of our protein organized into these other kind of smaller tube-looking structures, which are called myofibrils. Each individual myofibril, you can see here to here to there, is made up of these structures that we call sarcomeres. And a myofibril is a sarcomere with another sarcomere adjacent to it, and another, and another, and another. And so because of that, the myofibril has this very distinct striped pattern that is going to look like under a microscope. We call that a striation. Okay? Skeletal muscle has striation. It's a very organized. Cardiac muscle also has striation. The smooth muscle that surrounds, say, your digestive system, or the smooth muscle that surrounds the arterioles in your cardiovascular system, doesn't look like this. We'll talk about those later. Okay? But it has this very characteristic striping. That characteristic striping comes from the organization and the pattern of these little small filaments that are made up of actin and myosin proteins. Okay? Myosin is a bigger protein than actin. And so its filaments, when you stack myosin next to each other, they look thick or thicker. They look darker under a microscope. And I'll show you guys a couple of pictures of that. Okay? They look a certain way. These actin filaments are going to be adjacent to them, but they're smaller. We call them thin filaments. And so an individual sarcomere is going to have these myosin filaments with 6 actin filaments around it, another myosin filament, 6 actin filaments kind of in 3 dimensions all around it, and then they're going to be stacked. Okay? So if you look, a myosin filament is going to be made up of these myosin molecules that, to be honest, kind of look like spark. Okay? They're going to have a head, and they're going to have a tail, and in the filaments, the tails are going to form this helix and wrap themselves around each other. Okay? The head of the myosin This is where force generation comes from. This is where ATP use comes from, okay? So the function of muscles come from what happens right here in the myosin head, okay? Actin molecules, each actin is a sphere, a little ball. And so a thin actin filament is going to be sphere, make a rope basically out of these little balls, and you take two of those and you wind them around each other, and they look that way. There's a couple of regulatory proteins that exist on the actin filament, which we will talk about, but they're smaller, they're thinner. A myosin filament, I think I have a picture of this, but I will draw it anyway. But imagine that I'm going to take this sarcomere, and we're going to turn it so that you're looking down into it. Okay, you're looking into it, and in the middle, you have a myosin filament, and coming off at these very particular angles inside the myosin filament are going to be the myosin heads. Okay, and then clustered around that is going to be then actin filament, actin filament, actin, 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 actin. Okay? And then over here, another thick filament, and another thin filament. And so you can imagine then what happens is there's this arrangement of 6 actin filaments making kind of a hexagon around every thick filament. So this arrangement, again, allows us to pack very tightly as much actin and myosin into as small of an area as possible. Okay? That's why they are arranged the way that they are. Okay? Everybody with me? Good. So there's kind of the sarcomeres. This is what sarcomeres look like. This is an electron micrograph of sarcomere. So what you can see then is this sarcomere. This is a sarcomere. This is a sarcomere. So they're all these very nice, tightly, easily organized structures that are going to be. You see the striping, okay? This one isn't even zoomed down as much, but if you kind of look, you feel like I can see the little filaments and things that are going to kind of be collected in here, okay? The middle portion is called an M line. This deal at the end is called the Z line or the Z disc. I think Dr. Mike makes the undergrads learn all of that. I don't care. Okay? It's in your book. You could look it up. It's not that important, other than we're going to see how the myosin and actin interact with each other. Okay? This is a better micrograph where you can see Z line here. This is the end, this is an end, here's the next one. So you notice how there's this lighter and then dark, and then lighter and then dark. What's happening is in this area in the middle, what's called the H band or the H zone, this is the area where I have both actin and myosin lined up next to each other. And then out here, where it's lighter, that's just actin, okay? So sarcomeres are going to have the thick filaments in the middle, and then there's going to be these actin filaments that are going to run from the middle out to the end, and they anchor themselves at this Z line, at the end of the sarcomere. And so what happens when you get a muscle contraction is all these myosin heads grab actin and they pull. The ones on this side pull things in this way, the ones on this side pull things in like this, and so the H band stays the same, but the Z lines are going to get moved slowly, slowly, slowly together, okay? So we're keeping the myosin in its sort of place, but we're pulling the actin filaments. And the actin filaments are anchored at the Z line so that when this part gets pulled, it pulls on this side. And then that pulls on over here, and so on and so forth, and it helps us to kind of transmit our force along. Okay, there's the arrangement. You can see what that looks like. Let me give you a couple of more details about what's going on in the myosin head, especially at the end of the myosin filaments, because this is going to be where the force is generated, where the ATP is used. So this is really, really important from a structural standpoint. Okay, so myosin filaments are made up of what's called light myromyosin and heavy myromyosin. Okay, light myromyosin is going to form these tail structures. Okay, it is going to have two intertwined filaments that are made up of what we call the myosin heavy chain. Heavy just means it has a larger molecular weight than the light chain does. Okay, heavy myromyosin, not to be confused with the heavy chain. This is the piece, okay, that makes the globular heads, and this is going to be the piece of the myosin that will come into contact with the actin and form what we call a cross bridge. Okay, so what you can see here is this portion down here, this is the tail region. So this would be light myromyosin, made up of these individual molecules of things that we call the myosin heavy chain. Okay, myosin heavy chain will be very, very important in just a minute. Okay, it's going to then run the heavy chain up, run the heavy chain up. This is going to be one of the head regions. Here's the other head region. In the head region, there's some heavy chain molecules. There's going to be these different, what we call light chain myosin molecules. Again, they're just smaller. They have a smaller molecular weight. That's why they are called light chain. It denotes their weight, okay? And then at the end, there's the part that binds to the actin. There's an enzyme called an ATPase, okay? So this here is what forms our cross bridge. And then this ATPase enzyme, this is where all of your energy is from doing physical activity and movement every day comes from. It comes from this enzyme breaking down ATP. Okay? So it's going to connect to the actin, it's going to move, and the ATPase is going to hydrolyze ATP, it's going to let go, it's going to go back to its original conformational shape, and the whole thing starts over. Okay? So, heavy chain and the individual light chains and the ATPase enzyme are going to vary based upon your individual muscle fiber type. Okay? We'll talk about fiber types kind of at the end of all of this, but some muscle fibers or some myosin molecules have an ATPase that goes faster than others. It's what we call fast twitch muscles. People that are good sprinters have a faster ATPase enzyme. They have a slightly different myosin heavy chain. We know this because we can have a molecular, we can do an antibody target, we use something called a Western blot. We can target the heavy chain with an antibody, and it will tell us then what kind of myosin is this. And so it's called the MHC. So we can do that. The light chains are mostly regulatory. They help the function of the binding. They help the function of the enzyme. There's a few things that go on. We can phosphorylate the light chain. We do something called post-activation potentiation, and things is probably controlled by the light chain molecules are going to be here. But a lot of that's probably just a little beyond the scope of this class. Okay. All right. Questions? You guys still with me? Let's see what the next slide is. Let's talk about troponin and tropomyosin really quickly, and we'll call it a day. Okay. So that's the heavy chain. We're now going to talk a little bit more so about what's going on the thin filament and the actin filament. Okay. I mentioned to you all earlier that on the actin filament, there are two regulatory proteins. Those are called tropomyosin and troponin. Okay? I like to make lots of toddler analogies now because I have a toddler and that sort of dominates here. But tropomyosin functions as the little plastic things that you stick in the light sockets to keep your toddler from being able to put their finger into the light socket. Okay? It's a protective shield, if you will. Tropomyosin is this long, rope-like structure, and it runs the length of the actin filament. But what it does is it sits in and covers up the binding site where myosin would want to bind on to actin. Okay? And so what that does then is the normal state of your muscles is to not be contracted. They're meant to be relaxed. They are relaxed because you have tropomyosin. If you didn't have tropomyosin, your entire body would be one big ass cramp, okay? Which would be both uncomfortable and metabolically very inefficient. Okay? So it sits in the actin binding site. So what you guys will figure out, I like to make this, I've got to tell the undergrads this because, I don't know, it just seems like an analogy that would make sense to them. Myosin is very creepy. It really likes actin. It's very affectionate. It's very handsy in many ways. It's kind of a creeper. It wants to grab that actin. It really, really badly wants to like grab it, give it a hug, whatever. Okay? We're now venturing into kind of weird territory. So we need something. We need the tropomyosin, right? To sit in the binding site and keep that actin from being able to grab onto the myosin from grabbing onto the actin. At various intervals along the filament, there's going to be another protein called troponin. Okay? Troponin has 3 subunits. It doesn't matter that it has 3 subunits. Okay? One of those subunits will bind calcium. It's called troponin C. But the point of the troponin is it's going to be interspersed. When that calcium that's released from the sarcoplasmic reticulum binds on that troponin, the troponin changes its shape. And it moves, and it's attached to the tropomyosin, but when it moves, it pulls the tropomyosin away from the actin binding site. Now, creepy myosin can grab on and do its thing. Okay? And so these are what we call them regulatory proteins. So the tropomyosin prevents binding when we don't want it, and then the troponin is sort of our ability when it gets the right signal, then it will allow the tropomyosin out of the way. Okay? So that's why we need the calcium from the sarcoplasmic reticulum, so that we can basically tell troponin to move and allow actin and myosin to interact when we want it to happen, not just whenever myosin wants to be creepy. Okay, we'll stop there for today. We'll stop there for today.